Vegetable fats and vegetable oils tend to have more unsaturated and polyunsaturated fats. Which one's better for you? Well, unsaturated fats are better for you. But if you're talking about engineered polyunsaturated fats like olestra, which was a thing back in the 80s, that was an engineered polyunsaturated fat that caused horrible diarrhea, greasy, fatty stools in people who ate it. Olestra chips, fat-free potato chips. What a wonderful concept, until you started pooping very large amounts of greasy, fatty stools. Because you couldn't process this fat, this polyunsaturated synthetic fat. Yeah. So, it's all about efficient packing. If they are bent, you cannot put bent things in an efficient order. They do not pack very well. It's like if you try and pack multiple spoons together, they either fit very perfectly or they're just complete crumbled mess. If they're not in a perfect straight line, they're just a complete mess. So that's the difference between unsaturated and saturated from that melting point perspective. How densely do they pack? So in terms of that, non-polar lipids, lipids that do not have a polar end, they are not amphipathic. They do not have a polar head. These are the waxes. Waxes are when you take two fatty acids and you stick them together by an ester bond, which is a condensation reaction between two carboxylic acid groups. It's still a condensation reaction. You are removing water and connecting them. The only difference is that is an ester bond. That is an ester bond. Steroids are also non-polar lipids. Steroids are recognized by their ring structure. They have a tetracyclic skeleton. 4 rings. Tetra, 4. Cyclic, circle. 4 circles. They have 3 fused 6-membered rings and a 5-membered ring. So if you see that shape, it is a steroid compound. You can turn a non-polar steroid into an amphipathic steroid by adding a hydroxyl group. That's all it takes, is you add a polar group and it is now amphipathic. This is triglyceride. This is also non-polar. It comes from 3 fatty acids and a glycerol. Glycerol, fatty acid tail 1, 2, 3. It came about by 3 condensation reactions. 1, 2, 3. 3 water molecules were lost. The OH was lost from this carbon over here. And the oxygen here remains on the glycerol. The hydrogen came from the glycerol. The glycerol is an alcohol. It is a proton donor in this instance. It gave up the hydrogen. Or, wait, no, I take it back. Glycerol, the alcohol, didn't give up the hydrogen. Fatty acid gave up the hydrogen. My apologies. The hydroxyl group came off of the glycerol. Good thing you don't need to use that on the Cambridge exam. I've never seen a question about triglyceride condensation reactions. It might show up. Who knows? Maybe this year's the charm. But chemistry is hard for me. Didn't seem that way early on, but chemistry is hard for me. The biology stuff later is much easier for me. So triglycerides are the main storage form of lipids in animals. And they are a fantastic energy source because of the carbon-carbon bonds. The carbon-carbon bonds are where all that energy is stored. Breaking that up is where you get the energy for metabolism and to produce ATP. It's why they burn so well. It's why oil is so flammable. It's because of all those carbon-carbon bonds. And because triglycerides are not soluble in water, they can be packed together into groups and just pushed out into one part. Adipocytes, fat cells, do this all the time. They create these big sacs of just triglyceride and just push them to one side and retain some small part of their cellular anatomy for their own metabolic functions and just have a big old storage of triglyceride just sitting there. And as you lose fat, you use up the triglyceride in these adipocytes, and the individual adipocytes become smaller. The number of adipocytes do not change. That is why it is so easy to get the fat back once you have lost it. The only way to make it easy to lose the fat and keep it off is to lose the weight and then get liposuction. That's the only easy way. Otherwise, you got to keep doing your diet forever. And who wants to do that? Who wants to eat right and be healthy forever?
Wait, what?
So, it's a condensation reaction. No, maybe I was right the first time. I was right the first time. I shouldn't have second guessed myself. The hydrogen is given up by the glycerol because alcohol is always the primary proton donor in any condensation reaction. Yeah. Sorry. Organic chemistry was hard. Organic chemistry is hard, you guys. All right? And that proceeds to the triglyceride, and you get 3H2O. That's it. So, this is a phospholipid. We take a polar group and we attach it to the glycerol and leave the fatty acid tails alone. So now we have a polar double tail. 2 fatty acid tails and a phosphocholine hydrophilic head. A positive and negative charge here. Overall, this is neutrally charged, but because it is a positive and a negative side, because it is a dipole, it is polar. This is polar, and this is non-polar. This is what gives us our lipid bilayer. And this is how we turn cholesterol into an antipathic lipid. We just add a hydroxyl group. This is a steroid compound. These are just R groups, doesn't matter what they are really. This is just cholesterol. That hydroxyl group right there makes it amphipathic. That is the polar head. That is what lets it float in the lipid bilayer. So we end up with hydroxyl group here, carbon ring structure here, and whatever tail goes in here. This is oversized. This is not to scale. These phospholipids are much, much larger and much, much bigger than the cholesterol. Cholesterol is tiny and just stuck in as a peg. It is there to break up surface tension between the phosphorylcholine heads. It allows them to wiggle more. It makes them more fluid, so that when the temperature goes down and you hit it with a hammer, it doesn't crack. It ripples. It bends as the willow instead of snapping like the oak tree. Very kung fu of it. So that was when we did this in class. That was craziness. This is the amino acid chains. So the amino acid stuff. We're on amino acids now. Now we're on amino acids. We are more than halfway through Unit 1. The amino acid group. The amino group is the NH2. The variable side chain is the R group. The carboxylic acid group is the C double bond O, OH. And the alpha carbon is the center carbon around which everything else revolves, and there is always a hydrogen attached to it. Otherwise, it's not really an amino acid. Then it's something else. Then it's weird. This R group could be hydrogen. And then it is the simplest amino acid. And I forget whether or not that's glycine or something. Yeah, glycine. So this is the structure of the amino acid. This is its anatomy. The amino group is a proton acceptor, a Bronsted-Lowry base. The carboxylic acid group is a proton donor, a Bronsted-Lowry acid. It donates a proton, the hydrogen. This accepts a proton, accepts a new hydrogen. It becomes NH3 plus, and this becomes COO minus. This R chain, who cares what it does? We don't. We don't care until PhD level. And you are not there. One step at a time. So, because it does these things, it converts. We always list it as accepting the proton on this side and we're donating the proton on this side before we do any kind of reaction. Because otherwise, if it were neutral, it doesn't matter what arrangement they're in. A carboxylic acid could bind with a carboxylic acid and we would get an ester bond. And an ester bond is no good to us. That's not a peptide bond. But because one is a proton acceptor and the other is a proton donor, they have charge. They lose a proton, gain a proton, negative, positive. They become little bar magnets and they line up end to end, COO minus to NH3 plus. That is how they always line up. They are always lined up in that orientation because of their charge, because of their electrical charge, because of their electrical orientation, their dipole moment. And I've already shown you in the first hour how you draw this part of it. You draw a circle around the O minus and two of the H pluses and two of the H's with the H2O leaving. And you end up with whatever you had here and whatever you had here with the peptide bond. CO and H, up, down. Up, down, because otherwise they just get in the way and it's ugly, but also for organic chemistry reasons that you don't understand yet. Because you don't need to know it. You don't need it. And this is not chemistry. This is not chemistry. This is biology. So it's not in your pay. It's not your pay grade right now. But the final results of the reaction are this plus H2O. Plus H2O. Showing the intermediate step here would be circling the two H's and the O minus and showing water leaving. This is the final reaction step. So protein structures, the levels of protein structure, we need to go over this. Primary structure is the sequence of amino acids. This is an amino acid of certain R group. This is an amino acid with a certain R group. Each one has either a different R group or the same R group. There's the same configuration. That it is a chain isn't the primary structure. It is the sequence of individual amino acids. That it is glycine, leucine, isoleucine. Not that they are in a chain. Which ones are they and what order are they in? That is the primary structure. What is the order of amino acids? That is the key. That is what is primary structure. So each one has an identity in the primary structure. Each of these individual amino acids has its own identity, and that never changes, as determined by the DNA and post-translational modification. The relevant bonds here. Hydrogen bonding is relevant. Hydrogen to oxygen. The electronegative to the electropositive. The positively charged hydrogen, the negatively charged oxygen. They are forming a dipole, and they are like little bar magnets getting in orientation to one another. This is a hydrogen bond denoted by a dashed line. When you see a dashed line between two things, that is a hydrogen bond. That is 3 hydrogen bonds. That is 2 hydrogen bonds. How many dashed lines? How many hydrogen bonds? Why did I draw A and T and G and C? Unit 3. Hydrogen bonds are dashed lines. Covalent bonds are solid lines. CONH. That's a peptide bond. Whatever else is over here, etc. But it's a covalent bond. Solid lines are covalent. Dotted lines are hydrogen bonds. Those are relevant. They are relevant to secondary structure. The 2 types of secondary structure are beta pleated sheets and alpha helices. The alpha helix is a spiral shape where the hydrogens are interacting with the oxygens, and the carboxylic acid double bond O oxygens and the nitrogenous and the amine group hydrogens. They're interacting with each other with neighbors from other, like nearby neighbors, from other, not right next door to each other. And that forms this alpha helix. These dots are hydrogen bonds. Beta pleated sheets are pleats. They are like those compact ribbons, accordions that you unfurl. And they are hydrogen bonds between the same things, the amine group and the carboxylic acid oxygen. The amine group hydrogen and the carboxylic group oxygen, but from neighbors far across the divide, not nearby each other. They just are on opposite streets. So pleated sheets alpha helices. Those are your two forms of secondary structure. Both require hydrogen bonds. Both rely 100% on hydrogen bonds. It is the only relevant bond involved in secondary structure. The relevant bonds for the next level of structure are ionic bonds, where the electron is taken, not shared. Or a disulfide bond, where you have a covalent bond between two sulfurs. And you end up with tertiary structure. Tertiary, the third level of protein structure. You also have hydrogen bonds in play here. They are also important. You also have hydrophilic and hydrophobic interactions in play. Hydrophilic groups like to stick together and like to stick to the aqueous medium surrounding, if we're talking about a globular protein. Hydrophobic molecules like to stick together and avoid water and other polar compounds. So in a globular protein, in a protein that has a ball shape, hydrophobic stuff will tend to be on the inside, and hydrophilic stuff will tend to be on the outside. If you're talking about a protein that protrudes through the cellular membrane, the hydrophilic stuff will face the outside and inside of the cell, and the hydrophobic stuff will face the intermembrane, the interbilayer space where the hydrophobic tails are, where the long fatty acid chains are that make up the hydrophobic tails of the phospholipids. And then hydrophobic will interact only with hydrophobic. So that determines tertiary structure. That is hydrophilic and hydrophobic interactions. Disulfide bonds are your strongest bond here, because ionic bonds, salt bridges, can be broken in an aqueous solution. That's why ionic bonds are not strong in biology. Even though in chemistry they are the strongest bonds, they are the hardest to break, in biology they are the easiest to break because you just add water and they're gone. The polarity is what keeps them strong. And when you put them in a polar medium, they are not strong anymore. Now they are distracted by all the fancy shiny water around them. So this tertiary structure includes beta-pleated sheets. It includes alpha helices within its structure. Secondary structure combines with these additional interactions to form the 3D conformation. Tertiary structure, third level structure, 3D structure. It is the 3D structure of the protein. You only need tertiary structure to have a functional protein. You do not need to go further, but you can. Nope, stop that. A quaternary structure is the final level of structure. There is nothing beyond this. Quaternary structure is when you have more than one tertiary structure. You simply take two 3D things and you combine them and you link them together somehow, either through non-peptide bonding or just through shape linking. You make them hook together somehow. And you end up with, a good example is hemoglobin, where you have 4 chains. Another good example would be Antibodies. Antibodies are a good example, where you have more than one chain linked together by disulfide bridges. There are 1, 2, 3, 4 chains here as well. Those are, and sometimes you can have, you can also have the inactivated form of insulin, which is a 6 insulin structure connected together and bound by disulfide bridges. When you break those disulfide bridges, the whole thing comes apart and you have 6 separate insulin molecules. The 6 separate individual protein chains themselves are functional proteins with a 3D conformation. It just takes more than one chain. That's it. You don't need 4 chains to make a quaternary structure. You just need more than one. More than one. And I think that's actually where we'll cut this round. So you have another 5-minute break.